So unlike the other units, this one I feel like we really kind of hit pretty hard, especially with the different steps of like cell respiration and photosynthesis. So I'm going to just kind of remind you what's in it, give you kind of the high points of it, and then I'm not going to redraw the entire process, okay? Because I think we have that drawn somewhere, like multiple times. And like if you're like not sure about that, I mean, I still got these. I still got these little sheets that you can practice with. There's a whole stack of them up here. So with both sides, and you can play with that if you want to. But it just is like, okay, we're getting to the point where it's like that's going to be over the top, too much stuff to put on one little review sheet. So though, after, I reviewed all the standards yesterday just to kind of make sure I was getting everything. You do have to know it, like all that stuff that that's on that sheet, like it's not optional. It's word for word in your standards. Like this does that and this does that. So we can't just be like, oh, well, that's not going to be on there. In fact, what I'm hopeful for today is that I can finish this up in decent time. I can return your mocks to you, give you 15 minutes or something at the end. We can pull it out. We can look at FRQ1 because that had a chunk of cellular respiration in it. It was all about NADH and NAD plus, and look at how we could have used this information to answer that question appropriately. So that's kind of what I'm hopeful for. So that being said, I'm going to try to buzz through this pretty quickly. Okay. There's three main parts of energy. And so I'm going to have like one part here, and then a chunk in the middle. And that's going to be a little bit, it's still, to me, my little pretend drawings that I was doing are a little bit crowded, but we'll try to figure it out. So the three parts, enzymes is the first part. So enzymes going to go in the middle. Enzymes are going to be like, the things that actually lower the amount of energy that the cells require to do these things. And sometimes it won't even exist, like the process won't happen without an enzyme. But at the very, very least, they make it easier to happen. Okay, so enzymes are important in energy because it's energy conservation. It's helping that reaction happen faster. And then, of course, the other two are photosynthesis and cell respiration. Those are our big energy processes. Okay, so I'm going to do my best to kind of gloss over it. It's actually really difficult for me to gloss. I have a tendency to want to dive into details, and it's like, okay, we don't need that on a review sheet. We have that, somewhere we can look it up. So, okay, so we're going to just start kind of by listing the little on the top by the name here. I've got just a couple little facts that they want you to know. And basically, organisms have to have more input of energy than output. And that makes sense, right? Because much of energy is lost as heat whenever processes occur. Okay, so it's not like they're able to conserve all of their energy for the process. So that's kind of an important thing that they want you to know. Hey, the organism has to acquire more energy that it's going to need, that it's going to use for its processes, because every process isn't 100% efficient. And so you're losing some of the energy that would be required. So that's kind of one thing they want to know. They also wanted you to know over here on this side, I'm going to talk about photosynthesis a little bit, that photosynthesis, evolutionarily speaking, is considered the first of the two processes. So I'm going to say photosynthesis was first. Prokaryotes began. So prokaryotes started at, oh gosh, got hair where. Started, and I'm talking about photosynthesis here. So photosynthesis was first, the prokaryotes started, and that oxygenated the atmosphere. I don't know how to exactly say this in a few words. So created oxygen in atmosphere. Okay, so that's kind of an evolutionary part. Not that that's a heavily tested thing, but it is in your standards, just as far as knowing, hey, they think that photosynthesis actually was the first of the two processes to get going, and that created a situation where oxygen was released. And so by that release of oxygen, the atmosphere became something that we can sort of recognize today. There you go. Okay. All right. So let's start with enzymes in the middle here. And so here's our first of our three. Okay. Enzymes was a little bit hard for us in class. Like that enzyme quiz, like ate your lunch multiple times. It was like a little bully. Sorry about that. But so let's just kind of hit the high points here and make sure you know. Enzymes are which class of macromolecule? Protein. Enzymes are a protein. So they have a tertiary shape, right? So on enzymes, basically what you have is you have a structure. I'm just going to draw a funky looking little enzyme. You know, they're kind of like a blob. You have a structure that has an active site. And so this looks like a blob, but really, you know, it's a long chain of amino acids that's wadded up. So you can think of it like, I don't know, a wad of dental floss or something. So if I was going to draw it, you know, it'd be like a little string of stuff that's all wadded up. If I wanted it to kind of look like an enzyme would. And that's literally what they look like. They look like a ball of stuff, you know, and they have some coils in them because they have some secondary structure and stuff like that. But what we've got here is we have an active site. And so that's important. If you don't know that the active site is there because of the folding of it, like the shape of it, then make yourself a note. Like this happens because it's folded a certain way, right? Does everyone okay with that? So if it's folded a certain way, if you get a mutation, it folds a different way. Could be, potentially, right? If it's a weird mutation, potentially it could fold differently. If it folds differently, what happens? It changes the shape. And if that affects the active site, now it doesn't work anymore, or it doesn't work as well. Okay, so it might work sort of, but not very efficiently. Like it has to work at it. And so then you would have a substrate. So I'm going to kind of draw a little substrate over here. So if it goes down in here, that fits in, and we're gonna call it a substrate. So you know that it's what it is. Okay. If you ever see like substrate level something, like substrate level phosphorylation, substrate level, there's a couple of different times that they can pop that term up. Just know that it's talking about something that, you know, like if it's phosphorylating, it has to have like a substrate involved. So there has to be some enzyme activity with anything that's substrate level. Okay. Okay. I'll color him in. So here's our little substrate, and you'll notice that it fits nicely. Okay. And so what do you need to know about that? Well, obviously we already talked about it mutates, but there are some ways to inhibit it, right? So you can inhibit the function of the enzyme by competitive. What is a competitive inhibitor? Like, how does that work? Yeah.
Well, it just goes into the active site.
Right. Your competitive inhibitor is going to sit down in here and be like, hey, I stole your active site. I beat you. We were competing and I won, right? And so a competitive inhibitor basically just blocks your active site. Okay, so competitive is going to be blocks active site. Okay. All right, so then the other one is called a non-competitive or allosteric. I'm going to write allosteric. They'll say non-competitive. They'll say allosteric. You never know which they're going to say. Just know there's only two, so it's not like there's three kinds: allosteric, non-competitive, and you know, whatever, and competitive. No, there's just two kinds. So allosteric, if you're worried about that, you can put non-competitor under it. And what does it do? How does an allosteric inhibitor work? If it doesn't block the active site, how does it inhibit? You remember?
It attaches to another part of the enzyme.
Right, so maybe we have an allosteric site right here. Maybe this right here, we're going to call it the allosteric site, okay? Just a little bump in it. Find a bump in here somewhere. So this is the allosteric site, okay? So this one changes the shape of the enzyme. Okay, I called it an ENZY. I'll give it a new name. Get a nickname. So allosteric locks on somewhere else, and because it is a chain of amino acids and the R groups have properties, right? When that allosteric inhibitor binds there, it causes a chain reaction, if you will, that refolds it in such a way that the active site isn't the correct shape for your substrate anymore, and now it doesn't work. Allosteric inhibitors are often the most inhibiting of the inhibitors because they actually make it where it's not like competition. Ooh, maybe your substrate will get there first, right? Could happen. Not like that anymore. Now it's like doors shut. Nobody works. So can you see how an allosteric inhibitor would create a situation where you have like no product made? Like whatever the substrate's turning into, there's not any because all the enzymes have been allosterically or non-competitively inhibited and they just can't anymore, okay? So you can also see like from our mock, if you had vitamin D or some kind of vitamin, there was one question that said, how can this vitamin increase the rate of enzyme activity when it's obviously not substrate, right? It's just shaped the same way. Well, that's because if you have more, say, non-inhibiting competitors that are around, it's basically like the vitamin would sit down in there and be like, oh, okay, now I'm gone. And it's almost like it's just keeping the competitor out, right? So it's like holding a spot for the substrate, and then it'll come and go. Okay, so that was kind of a deep thought that they wanted you to get. But remember, most of your questions are gonna have a deep thought somewhere in it, so that's okay too. So if you were like, I don't even know how adding vitamin to this is gonna help, that's okay too. But that was kind of the idea, was that it provides for more substrate, if you will. Okay. All right. So then at the bottom here, we need to finish out. We'll have a little bit of room here. We need to finish out like what changes it. So environmental effects, right? And you know these, I think. I hope, because we kind of went over them a lot whenever your quizzes didn't go well. And that was across the board in all my classes. So, okay. So how does the environment affect these little guys? Well, one, we can have temperature. So let's temperature. Now, hopefully temperature increases the rate of activity, right? Unless it gets too hot and then it denatures. Okay, so temperature is one. So an increase in temperature is an increase in activity because everything's sort of like vibrating around faster. And so, oh gosh, well, the substrate's going to be going in faster, right? So you're going to see it go like, oh, because temperature increases the like movement of a system. Just the fact that the movement is increased is going to increase your ability for the substrates to hit the active site just randomly. Okay. Cooling it down slows everything down. Okay. That's very typical. Now you get too hot though, and you can start denaturing things, which means denaturing means your enzyme's falling apart. And now your active site doesn't work anymore because your enzyme's not great. Okay. And pH is the one they use the most for denaturing. They talk about pH more than temperature as far as denaturing goes. You have an optimal range. And just know the optimal range is different for each enzyme, like depending on where its native range is. I feel like there was one other thing that I was going to mention on this. Can't remember what it was. Great. Oh well, we've got a spot for it if I come back for it. There's something else that's going to bug me. Okay. All right. So over here, we're going to highlight photosynthesis. Any questions about enzymes or anything that is glaringly that I missed? Because I did miss something. I just can't remember what it was. Yeah.
Does the concentration denature it?
Okay. Doesn't denature it, but that's a good, that might be what I was missing, Natalie. Concentration. Let's talk about that. And that's really low. I know that some of you guys aren't going to be able to see that. So number 3 says concentration. So you can do concentration of enzyme if you want to put a little branch off of it and say enzyme and substrate, okay? So you can do a concentration of enzyme, and a concentration of enzyme is going to increase. If you increase the concentration, it's going to increase your rate, okay? If you increase substrate, it's going to increase your rate more than increasing the enzyme because the enzyme's reusable, the substrate is not. So the enzymes, if you increase a bunch of enzymes, you're still gonna stop having product when you run out of substrate. If you increase your amount of substrate, you have the same amount of enzymes you did, but they can be reused. So when one substrate gets out, another one gets in. And so then you actually are gonna have more product from increasing your substrate than you would from increasing your enzyme. But both of them, if you increase concentration, will increase your products, okay? Yep.
Just for a reiteration, you said talking about the vitamin.
Yeah.
The vitamin will help the substrate.
In that particular instance, where they actually said in the question that the vitamin was the same shape and everything as the substrate. They're like, why is adding vitamin to it when you have a funky, sort of almost mutated, not working very well enzyme? Like, how does that help? Basically, it helps to like outcompete the competitor.
And what's a substrate then done? Because then it's all used.
Yeah, so a substrate would be like something you're trying to break apart or something you're trying to put together. So once you break it apart with your enzyme, then it's done. It's broken now and it's off to do whatever it needed to do. And so you don't need to do it again. Right now, there would be a second enzyme somewhere else that could put it back together, but not that same enzyme.
And so your R groups with your enzymes, so the enzymes, amino acids are What's in a substrate then?
A substrate could be a lot of different things. Like a substrate can be like hydrogen peroxide, or just whatever needs to be dealt with
Okay, yeah.
at the time. Yeah.
Is a vitamin like one of the cofactors of an enzyme?
So vitamins can be a cofactor of an enzyme, which basically just a cofactor just helps its productivity, which could be because of, like our question said, could be other reasons too. They don't really, cofactors are not in your standards. Okay, so I'm not putting it on here. Okay, any more questions on enzymes? All right, let's get to the familiar and comfortable, right? Photosynthesis. Okay, so photosynthesis over here, if you've got some space, all that's going to be in this column is photosynthesis, all the way to the bottom. So you can make, lines in between your enzyme here if you want to. Okay, so the first thing I want to do is just put the formula up here, and I'm going to do I'm going to do the short formula, not the long one. Make sure I'm still in my little video screen here. Yeah. So what we need 6. I'm going to do it in order of how we use them. 6 water come in and get split. Okay. And then in the Calvin cycle, we're going to need 6 CO2. And then that's going to yield, first thing we lose is our oxygen. So 6 oxygen and C6H12O6. Whew, barely fit. Okay. So there's our formula. You need to know, and I mean, you're probably not going to have to write the formula, and you're probably not going to have to do this next thing, but just, this should kind of be what you need to know. Basically, this is the light-dependent reaction. This happens in the Calvin cycle. This happens in the LDR, and this is the Calvin cycle, okay? So just know which part goes with which, but if you understand the process, you should know that. Like, it might take you a second to think about it. That's okay. You have a second to think about it. So it's not like you won't be able to sit there and consider, okay? So I'm going to draw a chloroplast. I'm going to draw it kind of big because I want you to be able to see a thylakoid. And this is an issue that we've had this issue too. I want just to make sure you know where you are. So this should be like the edge of your paper. So you only have half a chloroplast. And you should have a little space under it, but you don't need a ton. Because I drew this on the back of an envelope. That's what I had handy. So here's my original right here. Okay. Yeah, actually, I don't know that you need much space under it at all. Okay. All right. So you have to know what's like inside this thing. Like you have to know what things are called. This was on test 5, or was it 5? Yeah, unit 5 test, where it asked you where's the ETC? Actually, it called it P. It called it energy transfer proteins instead of it's called it electron transfer proteins instead of electron transfer chain. But if you put ETC, that was fine too. It's like, where is the ETC going to be? Where is the carbon fixation, or something like that, I think is what it said. Okay, so basically I realized at that point that we maybe weren't really sure where we were in the cell whenever I did my little paper thingies here. So this little paper thingy here, when I drew this little round thing, this is a thylakoid. Okay, this is not a chloroplast. This is a thylakoid, right? So it's zoomed in. And so some of you put this membrane was where the transport chain was. And it's actually this membrane, okay? So just to make sure, do you see the increase in surface area as opposed to if I just had one big oval thylakoid? Like if I made that one big thylakoid, do you see the increase in surface area of having a bunch of little ones, more membrane, lots of membrane? Yeah. It's one of your kind of organelle things. These are almost like organelles inside an organelle, right? So kind of crazy. All right. So that was kind of the deal there. So that was, yeah, I feel like I didn't do a great job there. Sorry about that. Okay. So we have stroma here. It's just like the jelly stuff. And we have thylakoid here. Oh, wow. I'm just going to squish stuff. Sorry about that. And then one of these is called a grana. Once again, grana or granum. Granum is plural, grana is singular. Doesn't matter if you know that or not. Just call it a grana or granum. Who cares? It's so basically this is the thing they stick in their standards, and I'm like, why do you call it that and not just that? I don't know why we care what a stack of them is called. Okay, so I'm going to draw I'm just going to put a little bit of protein on here. Just like pretend like we're doing our thing, only I'm just going to draw it little tiny and we're not going to go through every step. Okay. I just want the big picture right now. And then I'm going to put my ATP synthase here. It's like, looks like a little hot air balloon. Okay. Whoops. Hang on. I got to get my hot air balloon going the right way. Okay. So I'm not going to go, okay, we're going to go water is split and then the electrons go up in the PS2, and I'm not going to step through every bit of it for you until we're finished and we're working with the free response question from your mock, and then we'll go through it and make sure that we understand all the parts. But for your little thing, basically what I want you to write on your little paper, if you hopefully have the space, is I want you to put like, what do we get out of the light dependent reactions? Like, what is the end result? And what happens? Like what's going in, what's going out, and then where's it going, kind of thing. Okay. So I'm going to put my Calvin cycle here in the stroma. So I'm just going to, if you don't know Calvin cycle, write it out. I'm going to write CC, and I'm going to draw a little circle that represents a cycle. Okay. This cycle doesn't actually dance around in a circle. It's all staying still. But I'm going to go ahead and just make it like a little circle. Okay. Calvin cycle's in the stroma, and these are LDR. If you don't know what that is, write it out somewhere. You can make yourself a little key down here if you need to. Light-dependent reactions, Calvin cycle. Okay, so now you can start kind of plugging in stuff. So water goes in, but really what I care about, is that you have a little arrow that says what's going to the Calvin cycle from here and what is going to the Calvin cycle from the light-dependent reaction. Carbon's coming into the Calvin cycle from outside. Okay, but what does the light-dependent reactions create that powers the Calvin cycle? Like what? We have ATP coming over, so write ATP on there somewhere. And what else? What's our carrier? Electron carriers. Which one is the photosynthesis electron carrier? NADPH, right? So I would just put a little arrow saying, hey, from here we get NADPH. Oh my gosh, so tiny and cute. From here, we get NADPH leaving and going to the Calvin cycle, and ATP leaving and going to the Calvin cycle, and that powers the Calvin cycle. Okay. You need to know every step of the light-dependent reactions. You do not need to know every step of the Calvin cycle. So light-dependent reactions, it lists step by step in your standards. This happens first, then this, and it's exactly what we did in class. Water splitting, electrons going to PS2. Electrons go down the electron transport chain, powers the hydrogen or the proton gradient, powers the protons coming in, so you create a gradient in the inside of the thylakoid, right? You remember all that stuff, right? And even though I said 100 times, and here's 101, they love to talk about the proton gradient inside the thylakoid. They love it. They love to talk about leaky thylakoid membranes and how it'll lose the protons, and now, oh my gosh, no ATP. Because you don't have any protons to go through the ATP synthase. They adore that. I don't know why. It was on a practice test. We got it right at about 54%. So they got us again, dang it. Okay, so make sure that you know that that's going to be potentially something you see, because I have seen it over and over again. They love to talk about the proton gradient that builds up inside this little thylakoid that's supposed to go through the ATP synthase. But apparently, there's a lot of stuff out there that can screw up the thylakoid membrane and make that not happen. Okay, so they have all kinds of case studies, chemicals, drugs, mutations, you name it. And your thylakoid membrane doesn't hold, and the protons escape out other places, and you get no or little ATP. I don't know why that's the poster child, but it is. Okay, so make sure that you just kind of keep that in mind. All right, so carbon fixation occurs here in the form of carbon dioxide. So I'm going to put a little arrow into it and say carbon fixing, because I've seen that multiple times, and that's all you need to know. Besides that glucose comes out the other side, and I know we did G3P because we're awesome. It takes 3 G3P to make a glucose, but we'll just write glucose because that's what your standard says. Okay? So we'll skip a chunk. So what this says, because it's teeny tiny and you probably can't see it, is the LDR occurs on the thylakoid membrane. That's your electron transport chain, right? Right there in the middle. Okay? So your LDR occurs there. You're going to get energy from that, and it's going to fuel the Calvin cycle, which will fix carbon into glucose. That's what I want on your review sheet. Okay? Now, when you need to get deeper, but you can go back into your notes and get deeper than that, okay? And I would definitely review that before the test sometime, but just know that the overall picture will get you quite a bit, okay? Okay, questions on photosynthesis? Any questions? Photosynthesis is, I mean, obviously it's super duper important. If you got the because otherwise we'd all be dead, so it's kind of important. But you got the overall kind of picture of it, and then you can talk about kind of the evolution of it, right? Because, you know, our organisms, what does photosynthesis, by the way? Which organisms? The autotrophs do, and we have some, okay, so this would be a eukaryotic autotroph, right? Because it has a chloroplast. So this would be a plant, possibly an algae of some sort, right? Those are eukaryotes. We also have photosynthetic bacteria that just do this on folds of their membrane. They don't have chloroplast, okay? So they have to just kind of mess with the folds on their membrane, okay? But we know we have them because they're the ones that started the whole thing and perhaps got engulfed and became an endosymbiotic thing, right? Okay, here's the vesicular membrane, and here's the original little membrane of it. Okay, just reviewing endosymbiotic theory. Okie dokie, let's look at our cell respiration. Okay, so these are coupled. These are coupled reactions, which happens a lot. Your photosynthesis and cell respiration. One is endothermic. Oh, geez, my little computer's gonna die. Better get a charger. One is endothermic, and one is exothermic. Where did my charger go? I have it in a little box somewhere. I swear I'd lose my head if it wasn't screwed on tight. Oh, here it is. I'm just thankful I knew where it was. That's pretty miracle. All right, so let's see. There we go. Okay. I'm just making sure we're still filming. Yeah, we are. Okay, so our coupled reactions, typically if you have one that creates energy or, you know, is binds up energy, you have a coupled reaction that does the opposite, okay? And in this case, we have a beautiful little coupled reaction because they use exactly, I mean, it's like the exact amount of energy this one kind of pulls from the sun and puts into a molecule, this one uses. And so they're like perfectly coupled. Some of them aren't quite that perfect, but all right, this one, you can put the formula on if you'd like right there. It's not difficult, you just flip it. Okay, so we start with the glucose. So that begins outside. Remember, this is going to happen in glycolysis, which is going to happen in the cytosol of the actual cell, right outside the organelle. And then later, we're going to need some oxygen, like way later, like at the very end. Okay, oops, I forgot the 6. And then from that, we're going to get all these carbons that you see here, these 6 carbons going to have to be released into the atmosphere. And then when we bind up oxygen with some of our hydrogens over there, then we're going to get some water. Okay? All right. So, and of course, we're going to form little molecules of ATP when we do this. So that's kind of the deal there. All right, so we are now going to be in, I mean, if you want to put your things in, we can. This is going to be in glycolysis. Oxygen is going to occur in the last phase. I guess we'll call it the ETC. There's no easy way to write that because it's the oxidative phosphorylation. I guess I could put like OP. That's kind of funny. CO2 comes in, leaves, this leaves in the Krebs cycle mostly. And then water leaves in the ETC. Okay, all right, so there's our stuffs. Okay, so what do we need to know? Well, first of all, we better know that it's a mitochondria. Okay, so let's draw out our little mitochondria. If you will, leave just a little bit of space underneath this one for us to draw our ATP. In the very end, we're going to sketch out an ATP and what the parts of it are. But otherwise, that's all you need. So draw your loopy-doopy high surface area inner membrane. Okay, so there's our mitochondria, if you want to label it. I'm sure you know what it is. Okay. And remember, these two both have their own DNA, right? So they will make their own proteins that are for their processes. Like if they need some electron transport proteins, they'll create those, or sometimes they'll create their own proteins that break down their NADH or whatever, which was the topic of question for response number 1. That was a mutation in a protein that broke down NADH, and some weird things happened. All right. So the first thing happens out here. I am going to just give you ins and outs. What is in, what is out? That's what we're going to do with this. Okay, what comes in, what comes out. I'm not going to do every wee little step. It just takes too long. Okay. So, and plus you have it a million times. So you get 2-pyruvate from that. You're going to get, I'm going to put ATP on the right and the electron carriers on the left of the arrow. So uses 2 ATP, creates 4. So net is 2. Overall, you get 2 ATP from one round of glycolysis. We're using one, oh, sorry. Do you know that this starts with glucose? Shoot. If you don't, put glucose somewhere on here. I should have put glycolysis and then put glucose under it. I'm going to do that. Sorry if I screwed up your notes, but if you know that or you want to put it out to the side or something, glycolysis is going to go up here. Whoa, what is this word I see? What is this word? Lysis. What the heck? What does that mean even? Have we had lysis every single day of review so far? Jeez. I'm going to start playing word search and find for lysis in biology. Okay, uses 2 ATP, creates 4 ATP, and also creates 4 NADH. Is it 4? I think it's 4. Okay. I think it's 2 per pyruvate. Okay, so that's what happens there. And then the pyruvate goes in here, and pyruvate becomes acetyl-CoA. I hate these words, they're way too long. Okay, which is going to feed the Krebs cycle, which is in the matrix of the mitochondria. If you don't know that, right? Matrix in that gooey area in the center. I'm going to turn acetyl-CoA into I'm going to put it right into a cycle. Notice I made it drop down into the cycle like that, if you want to, because it's not actually, it enters the cycle. The cycle's there. Okay? And so we're going to get a bunch of NADH. We're going to get FADH2. We're going to get a little bit of ATP. Just kind of write the stuff around it. I don't care where. I need you to know what we're getting. I don't care where you're getting it, as long as it's from the right step. I don't care if it's on the right, left, bottom of the circle, whatever. So it's got FADH2, NADH. You also get some NADH from your pyruvate thing here too, but don't worry too much about it. CO2 is going to leave right then. Bye, CO2. And of course, there are 6 total CO2, but hey, 3 per pyruvate, right? Because we had 2 pyruvate, we got to do this twice. Just see, here I go. I'm doing way too much information. I don't need to do all this right now. Okay, so let's put on our electron transport chain. It's going to be right here. Toss in some proteins. Going to try to hit the big part, which is coming up. And then here's your little hot air balloon guy, ATP synthase. Going to make the ATP. That's what we're going for. Okay, and you know that the electrons go through, okay, so write ETC above it if you need to, electron transport chain. And this is your ATP synthase. Altogether, this is the oxidative phosphorylation. I'm going to call it OP.
Can you go over fermentation too?
Yeah, I will. That's going to be right after I'm finished here. Okay, so electron transport chain, your electrons go down, but here's the important part. Oxygen comes in. This is what we needed to remember with our free response questions, very frequently asked as the final electron acceptor. In a second, I'm going to highlight what I've seen asked over and over again, and then water comes out of that. Okay, and I'm not putting numbers on there. I don't care. And then, of course, then the ATP is created in lots of ATP. It's created by tons inside that ATP synthase. Okay, so you'll see the questions about glycolysis because it's in every single organism that exists, and so you'll see questions about glycolysis. I haven't seen much about the Krebs cycle. Like every once in a while, something will pop in, but not much. I have seen a ton about oxygen being the final electron acceptor. Like if I had to say what they love to talk about, I've already told you about the thylakoid, right? The thylakoid, leaky thylakoid, they love that one. And they love to talk about oxygen as your final electron acceptor. You'll see it asked in a lot of different ways. And I think it's because a lot of different mutations of proteins can change that. And so they have a lot of material to use because they don't just make stuff up on the AP test. You know, it has to be like actual things that happen in biology. And so there's just a lot of studies, a lot of material used for the proteins that are a part of the electron transport chain and that affect that oxygen use. And so you'll see those over and over again. It's just the way it is. Okay, so we need to make sure that that's in our brain. Like where is oxygen come into play? Like what's going to happen if your electron transport chain stops working or doesn't work as well? What's going to happen to your amount of oxygen if the electron transport chain isn't working? That's a good question. So the question asked what's going to happen to your oxygen consumption, like use, right? So if this isn't working, your consumption is going to decrease, right? It's going to just, and actually your oxygen levels would increase. They should be sitting there going, I mean, I guess I don't have any electrons to pick up today because there's none coming down the pike, you know, kind of thing. But they asked in the thing about use. We're going to look at that question if I'll shut up here in a minute. Okay. So questions about oxidative, the oxygen part, or what we call aerobic respiration, because it's using oxygen right here. That makes it aerobic. Any questions about that?
So what's going to happen with oxygen if the ETC isn't working?
So oxygen will just build up because you're not going to stop breathing. You know, I mean, that's kind of the thing. So it'll just keep building it up in the cells. You'll be doing glycolysis to make some ATP, but your oxygen's just going to be sitting there if there's something wrong. Usually we do glycolysis. We do lactic acid fermentation because we ran out of oxygen. But if there's something wrong with your proteins of your ETC, you'll flip to lactic acid fermentation, but your oxygen will just be like sitting there going, I'm just going to sit here because I don't have anything to do, and then more will come in, and you'll still be like nothing to do with it. Yep.
So whenever it just sits there, that's anaerobic?
So anaerobic respiration actually is a type of respiration that doesn't use oxygen. There's a wee little nuance between fermentation and anaerobic respiration. Fermentation is what we do whenever we don't have oxygen available for whatever reason. Usually it's because we've used it all up, we've running too hard and we've used all our oxygen. And so we'll flip over to fermentation. So here, let's do that, and then I'll talk about anaerobic respiration. Okay, so what happens if oxygen is like, no, I'm not here? Then that's like, well, then we're not going to send any more NADH because it's not being broken down. And so we're not going to make any more. And then it's like, oh gosh, well then don't send the pyruvate in. So all this is kind of a signaling thing. Don't send the pyruvate in because, we can't use it, so forget it. And so what happens then is I'm going to say lactic acid. It's the one that we do, but it could also be alcoholic. And so you want to make note, if this is confusing to you, you want to make note that this is when no oxygen is available. So somewhere in here, like this chunk only happens, this lactic acid fermentation only happens if it's no oxygen is available, okay? And otherwise, our cells will do regular old cell respiration because it is way more, produces way more ATP, and we can't really handle the lactic acid buildup in our cells, not for long periods of time. Yeasts, there are yeasts that do fermentation on purpose, even though oxygen is available, because they can handle the off-gassing of the alcohol, which creates like beer, wine, all kinds of stuff like that. They can handle that off-gassing of the alcohol. It doesn't poison their cells, and it's super fast. It's super energy efficient. Okay, so what happens then is that regenerates. So that takes NADH and regenerates it back into NAD+, which is what you have to run this again, right? So if you don't have NAD+, you can't run glycolysis again. And so then it can just sit there and until you have so much lactic acid you have to stop. Okay, kind of thing. Everyone cool with that? So that's just kind of a loop, a little tiny loop that'll go until you have too much lactic acid and you have to stop. Yep.
So it makes that just because you don't have oxygen.
It makes that when I don't have oxygen because it needs to have NAD plus in order to do glycolysis again. And so yes, that's only when there's not
So
oxygen.
you're just repeating glycolysis.
Mhm. Yeah, and lactic acid is just basically it's just a change in pyruvate. It breaks it down several stages. Now, the only difference between that and alcoholic fermentation is there's 2 steps in between pyruvate and alcohol, and there's only 1 step in between pyruvate and lactic acid. Okay, so that's the only difference. Okay, cool. Let's draw our ATP. Hopefully, you have a little square down here. I'm gonna draw it up, but I would, hopefully you have a little square right here to put your product there, and I'm gonna draw it up here somewhere so that you can see it better. Okay, so ATP, just really quick, what is ATP? It, of course, is your energy molecule. I'm trying to get, so we have 10 minutes to get you your test real quick. Okay, so ATP is basically RNA nucleotide. You have your ribose, so that's the sugar. You have what, I'm going to draw my weird little doggy looking thing, because it's adenine, right? So adenine, which is your 2-ring structure, your purine. And then instead of 1 phosphate, it has 3. Okay, so there's your phosphates. And they like to say that energy is stored there. And I put an arrow. Oh my gosh, energy. Spell, Miss Wilhite. Energy stored. Okay, so this is phosphate, 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 and energy is stored in the bond between those last 2 phosphates. Break 1 off, and you have diphosphate, right? ADP. And then put it back on, and you have ATP. That's it. That's our little buddy, ATP. Just like an RNA with a few extra phosphates. Questions?